What is really interesting to see now is that in the AMT-060 trial is that they have now evidence of relatively long-term expression of factor IX. So we're talking about 5% factor IX in the low-dose cohort patients and 7.5% factor IX on average in the high-dose cohort patients for more than 2 years now, almost 3 years even. And I think this is the second study that shows us relatively prolonged expression after the Natwani report that was published in 2014. In the Natwani New England Journal of Medicine paper, there was evidence of 2 to 5% factor IX, and now that shows that the expression is now sustained for already 8 years. So this is the second study now showing relatively prolonged expression of factor IX. I think what's interesting is that the levels are not going down despite the occasional emergence of transaminitis, which is always a concern, of course. If you have transaminitis, it may point to an adverse immune reaction that could clear the cells, but they found no evidence of such a reaction. The vector that was used in the AMT-060 trial and in the follow-up trial, the AMT-061 trial, is based on a particular serotype which is coined as AV5. It turns out that the incidence of pre-existing antibodies to AV5 is relatively low in the human population. And what's intriguing is that some of the patients that were enrolled in the trial had low titer anti-AAV5 pre-existing antibodies. And despite the presence of these low titer antibodies to AAV5, they still expressed factor IX after gene therapy. One could assume that the presence of even low titer antibodies against the vector particles would have neutralized these vector particles and abrogated gene transfer 100%. The data now show that this is not necessarily the case. What I found particularly intriguing was that one of the patients in cohort 1 that had the highest levels of factor IX, approximating 10%, had actually the highest titer of anti-AV5 antibodies, which isn't exactly what you would have expected. I think what with the AMT-060 was really important to assess the impact of changing serotypes. Let us not forget that the design of the AMT-060 vector is exactly the same as the vector that was published in the New England Journal of Medicine papers in 2011 and 2014 by the Natwani and Davidov group. But what's really different between these two different trials is that the capsid is different. Instead of using AV8, they use AV5. We don't fully understand all the repercussions of changing serotype for the moment, but I think in time we will get a better grasp of the biological consequences of these different serotypes on the efficacy and on the safety.